All right. Good afternoon, everyone. It is my pleasure to introduce Gabriel Oliveira. He will be talking about deep learning models for protein function prediction and beyond. He's a PhD candidate in the computer science department at the University of Campinas in Brazil. And he has been developing various machine learning models for large-scale biological data sets. And he will be telling us all about this today. All right, Gabriel, please go ahead.
Thank you, Professor Said, and thank you for the opportunity to present my research and present other topics too. So the title of the presentation is Deep Learning for Protein Function Prediction and Beyond. And this is the agenda of the presentation. So my name is Gabriel Bianchim de Oliveira. I'm from Brazil. I am a PhD candidate at University of Campinas in Brazil. And during my master's and my PhD, I researched about machine learning and bioinformatics, mainly applied for protein data. And also I worked on different tasks, for example, using biological and medical data. So my PhD work focus on protein function prediction. And this is the main point of the presentation today. So to start, it's important to understand why protein function analysis is important. So protein analysis is important to discover different aspects and applications, such as drug discovery and disease research. And also it's important to analyze the genetic and also to understand complex biological mechanisms. And considering the functions of protein, it usually divided into ontologies, and the main ontology applied to this problem is gene ontology. So gene ontology is divided into three ontologies that we can say like aspects about the function. The first one is biological process. So biological process indicates the biological process that each protein is involved in. The second one is cellular component. So cellular component, it's about the place that a protein plays specific function. And the last one is molecular function. That is about the molecular level of the function that each protein plays. So there is important characteristics about gene ontology. So gene ontology and biological process, cellular component, and molecular function is organized in a direct acyclic graph. So with that, we have specific functions that is more specific, and also we have like more general functions. In this case, more general are the superclasses. It is close to the root node of the ontology. And also we have the more specific that we can say subclasses of the superclasses ones. Another important point here is that if a protein has a specific function, it also has all the superclasses functions up to the root node. And also another important topic here is that a protein can have multiple functions and play different functions at the same time. So with that, when we're talking about protein function prediction using machine learning or classifiers, we are talking about multi-label classification problem. To illustrate the ontology organization, we have this figure. For example, we have molecular function in the root node. Molecular function has three children. The first one is binding. The second one is protein complex scaffold. The third one is transporter activity. And we have for binding the child protein binding, and protein binding has a child that is structural molecular activity. And considering structural molecular activity, it has two parents. The first one is protein binding, and the second one is protein complex scaffold. So if a protein plays, for example, structural molecular activity, it also plays all the functions up to the root node. So here we have in orange the function that a specific protein plays. It also has all the functions up to the root node in this example. So with that, there's a challenge about protein function analysis. The first challenge that we can highlight is that we have a small fraction of proteins with experimentally annotated functions because we can say that it's a high cost technique ranging from something close to 300 up to $30,000 per sequence. It depends on the technique applied to this analysis. And another important challenge here, and we can see that uh, there is a lot of proteins with the amino acid sequence available because we can see that it's like uh, low cost to sequence amino acid sequence from proteins. Something close to 200 up to $1,000 per sequence using, for example, mass spectrometry. And here we have a figure to illustrate the difference between proteins with this amino acid sequence available and the number of proteins with manually annotated functions. So in the left side, we have the number of proteins in UniProt KP over the years ranging from 2014 up to 2024. So in 2014, it is something close to 100 million proteins. And in the last year, it is something close to 250 million proteins. But in another, in the right side of the figure, we can see that the number of functions is quite different from the number of proteins sequenced. It's ranging from 250,000 proteins in 2014 up to something close to 1,500,000 proteins in the last year. So with this difference, we have the opportunities. So the opportunities is to apply computational methods to predict protein functions. And here we can focus mainly on models that use protein sequence because it's the most available information about proteins. We can divide the methods in two main groups. The first one is based on deep learning, and the second one is based on sequence alignments. And also we have another group that is an ensemble between these two groups, combining both strategies. And with that, we can improve the results of each one individually. So to understand sequence-based methods and deep learning-based methods, sequence alignment-based methods usually use local alignment tools. For example, BLAST and Diamond are two common tools applied for this task. And the main idea here is that proteins with similar sequence could have similar functions. But this kind of approach has some difficulties when dealing with a long sequence and also with sequence that has different sequence, a protein that has different sequence, but have the same functions that we say distant homology relationships. And on the other side, we have deep learning based models. So these models usually use neural networks uh, and more recently, transformers based methods. So transformers are the state-of-the-art approach for natural language processing, like ChatGPT. And with that, a lot of models have been developed using transformers pre-trained on protein data, so we can use this kind of approach to develop new methods using transformers. And here, transformers can learn contextual representations of the protein sequence. And with that, it can model better the more complex relationship between the sequence and the function compared to sequence alignment predictions. Okay, so the next point is about the methods in the literature. So we have methods in the literature that are developed to predict protein functions based on the protein sequence, but uh, it has two main limitations. The first one is that the methods in the literature cannot deal well with sequence with more than uh, 1000 amino acids usually. For example, the methods discard the remaining amino acids from the sequence or use some kind of truncation to deal with the sequence. And it's important, there is important case that proteins with more than 1000 amino acids, the function is played for a specific part far from the initial part. And another important limitation about the methods in the literature is about when they may then assemble between deep learning with sequence alignment tools, predictions. So the methods usually use a simple linear combination between the predictions. And it's important to analyze here what functions deep learning based methods and sequence alignment based methods have the better predictions. For example, you can give more weight for the predictions of deep learning based methods for a specific function that this kind of approach has better results and vice versa. So with that, in my research, we proposed a new method that we call SuperMago. And we also presented a new method that integrate that ensemble SuperMago, that is a transformer-based method, with local alignment tools, predictions, that we called SuperMago Plus. So I will start presenting SuperMago, that is the deep learning-based model. So this is the pipeline of SuperMago. So from the amino acid sequence, we pass through transformers models, two transformers models, to extract embeddings or features from the sequence. And after that, we pass through a layer classification. And in the end, we made an stacking or an ensemble between the predictions of the layer classification process. So I will discuss a little bit about each step of this pipeline. So the first one is embedding extraction. So for embedding extraction, we used two transformers model. The first one is ESM2T36. That is a model based on BERT, but pre-trained on protein data. And the second one is PROT5. And here PROT5 is an encoder-decoder architecture, but we use the encoder part to get the representations of the sequence. PROT5 is a T5 model pre-trained on protein data. And another important topic here is about the features. So during the extraction, the models get representations for each amino acid of the sequence. And to get the protein representation at all, we compute the mean representation across the amino acids. So with that, we have a final representation of the protein. But as I said, an important topic here is about large proteins. So these two models, ESM2 and PROT5, cannot deal with sequence with more than 1022 amino acids. So to classify proteins using the whole sequence, we split the sequence into subsequence with the sliding window technique without overlapping. And with each one of the subsequence, we get the embeddings from each backbone. And in the end, we aggregate the features to have a final representation of the protein considering all the subsequence of the amino acid sequence of the protein. So to do that, we compute the position-wise mean to have a final representation for each protein. Okay, so with that, we have five embedding from these two backbones. And the next part of the pipeline is layer classification. So with that, we train dependent classifiers for each one of these embeddings from different layers of each one of these two backbones. And the main idea here is that each classifier can capture different informations about the protein sequence. And in the end, it's important to aggregate the predictions to have a final output for this deep learning based method. So the last part is the stacking. So the main idea of the stacking is to aggregate the predictions of these 10 different classifiers. And with that, we can combine the predictions. And the idea to the combination about the predictions is the usage of a meta classifier to understand the weights for each one of the functions in the gene ontology ontologies. For example, here we have in a figure to illustrate this process. Here we have prediction from model 1 and term number 1, prediction for model 2 and prediction for model 1, term 2, and so on. And here we have prediction of model 2, term 1, prediction of model 2, term 2, and so on. So the idea is to train this meta classifier to find the weights for these connections. So with that, if model 1 is a good classifier for term 1, this weight is higher than this one. But in another case, if model 2 is the best model predictor for term 2, this weight has a higher weight than this one. And with that, we can improve the results compared to a simple linear combination between the predictions. So in the end of that process, we have deep learning based prediction. So next, we need a local alignment tool prediction. So the idea here is that proteins with similar sequence tend to share similar functions. And to make the predictions, we used Diamond. Diamond is a high-speed tool compared to BLAST. And during the alignments usually return a bit score that is a score about the alignment. And based on these scores, we uh, predicted using a weighting tool to combine the predictions considering these alignments. So with Supermago and Blast and Diamond prediction, to Supermago Plus, we need to combine the predictions again. And here, important topic is that Diamond may not have predictions to all proteins because in the training set that we use to compare the predictions in the evaluation set can have like different sequence, and with that, Diamond cannot align the sequence in the evaluation set with the training set. So with that, we adapted the meta classifier strategy. So we created a conditional. So if the Diamond can predict for a specific function, for a specific protein, so we train again the meta classifier with this condition. And in the case that Diamond cannot predict, we used only Supermago prediction. So here is the idea. If Diamond predict, we used this meta classifier to train these weights to each one of the scenarios for each function, considering Supermago and Diamond predictions. Okay, so in the end of this process, we have Supermago and Supermago Plus. The next point is about the dataset and the metrics. So to evaluate Supermago and compare with the literature, we used the dataset from Kappa Challenge. There is the main dataset about protein function prediction in the literature. More specifically, we used Kappa 5 that was released in 2023 as our benchmark. And to evaluate, we used the Fmax score. Fmax score combine precision and recall, considering different thresholds ranging from 0 up to 1 with step of 0. 0.01. And with precision and recall, uh, it is calculated the F1 score, and Fmax is the maximum value of F1 score considering all these thresholds. And here we have the results. So in the top part of the table, we have SuperMago, that our deep learning based model, compared uh, in comparison with the state-of-the-art methods like ProtGold, TameProt, PUGo, AtGo, and Tail. Both of these methods are based on transformers. And here we can see that our approach achieved the highest Fmax score for biological process, cellular component, and molecular function. We also evaluated using statistical methods, and our method in this scenario for deep learning based models achieved a statistically difference between other methods in the literature. In the bottom part of the table, we have SuperMago Plus, that is our ensemble of deep learning with local alignment tool, compare with different models in the literature, the state-of-the-art models in the literature, like Temprot Plus, PU-Go plus Diamond, AtGo Plus, and also Tail Plus. And here, our method achieved the highest F1 score for cellular component and molecular function, and also the second best result for biological process. Here again, we evaluated using statistical methods, and SuperMago Plus achieved presented difference, statistically difference between the other methods in the literature. So in the end of this, we also developed a web server. So in this web server, you can put your protein in a FASTA format, and you can access the results of our methods. So here we have our web server. So in this web server, you can put here your protein, like this one as an example. And here you can put like the minimal confidence of the predictions of the model. And with that, we can submit and take the results after something close to 5 minutes. The idea of the web server is that it's important here to use less resources than the SuperMago Plus method. For example, SuperMago Plus needs something close to 16 gigabytes to execute. And here we developed a method using less research. So we changed ESM2 T36 to ESM2 12. And with that, we also used quantization to reduce the memory. And in the end of this process, we reduced from 16 gigabytes up to something close to 300 megabytes of GPU usage. So this web server is running on a 5-year-old GPU, so it don't need a lot of research to run. And here we have the results. So here we have the table of the results. Here is the protein name, the protein sequence that we sent to this web server. Here we can see the results for biological process, cellular component, and molecular function. For example, for biological process, considering the confidence, the minimum confidence of 0.5, this is the functions that our model predicted. And here also it has a figure to illustrate. So this is the root node, and this is the predictions. So we have like this node with in a green. So green means that the score, the confidence of the model is more than 0.75 for this function. And yellow, we have functions between 0.5 and 0.75. Here are the results for biological process, and we have also for cellular component here and the figure, and also molecular function here and the figure. And an interesting thing about this web server is that you can download the results, for example, in a JSON format or a CSV format. And also you can download all the formats and the figures too. So in the end, this is our web server about SuperMago Plus method. Okay, so now I will talk about other projects that I worked during my master's and my PhD. So there is three main points here about the projects. The first one is protein secondary structure prediction. The second one is COVID-19 classification. And the last one is medical and biological data applications. So protein secondary structure prediction is the research in I worked on my master's. And here the challenge is quite similar with protein function prediction. So there is a difference between the number of proteins, sequence proteins, and proteins with known structures. And the main idea is that based on the amino acid sequence, to predict the three-dimensional structure of each one of the amino acids of the sequence. And here again, the methods is divided into two approaches. The first one is based on deep learning, and the second one is based on sequence similarity, and in this case, local alignment tools. So in my master's, the final paper that we developed in my master's, we created a model using different deep learning models ranging from convolutional neural networks. And here in this point, we transform the protein sequence into images and use convolutional neural networks to classify images based on the amino acid sequence. The second one is LSTM-based models. So here we developed LSTM models to deal the sequence as a time series. We also used classical machine learning classifiers like random forest. Here we extracted features from the amino acid sequence and use this kind of classifier. And the last one is transformers. So here in that point, uh, the, the transformers pre-trained on protein data were not available at that point. So here we used transformers pre-trained on English language to classify protein functions. And after that, we evaluated more recently transformers for pre-trained on protein data, and the result is better with with transformers pre-trained on protein data. And we also used BLAST to make predictions using sequence similarity. And in the end of this process, we have two classifiers, one based on deep learning and another based on local alignment predictions. And in the end, we combine again these two approaches to have a final prediction. And the result was interesting. We compare our results with the literature, and also we achieved the state-of-the-art results in some scenarios. Another important project that I worked on is about COVID-19 classification. So this project uh, we developed during my master's and the beginning of my PhD. We evaluated different kind of data ranging from X-ray and CT scans. And here, based on these two data types, we applied convolutional neural networks to classify based on the image extracted from these two data. And the main focus of this project is about preprocessing techniques to evaluate how the preprocessing steps can help and can indicate the correct part of the image that the model should pay attention. And also another important topic here is about interpretability. So we evaluated what kind of what part of the image the model is paying attention and if it's correct considering this kind of task. So here are the classification use X-ray images. So based on the original image, like this one, we extracted 90 different patches from these images. So here in the left side, we have the five first patches, these five parts here, and also the central one. And we also extracted more four patches, that is this part on the right side of the figure. So with that, in the end of this process, we pass these 9 different parts of the image in a convolutional neural network to classify, and in the end, we ensemble the predictions of the convolutional neural network models. And here is an interesting image. So we focus to understand what kind of what part of the images the model pay attention. So here we can see in the red part of the figure that usually the model pay attention on the lung part of the patient. So it's important to understand the model predictions when we are working with medical or biological data. And here is about CT scans. And again, we focus on preprocessing steps about the images. So from the original image, like this one, we used different filters to identify specific regions of the images. And in the end of the process, the main point here is to identify the interior chest and the exterior chest of the images. So with that, based on this original image here, in the end, we have these two images, like exterior and interior chest, after applying this preprocessing function. And with these images, we also pass through convolutional neural networks. And in the end, we have the predictions. And here are the results about interpretability. So the data set is divided into three classes: COVID pneumonia, non-COVID pneumonia, like bacterial pneumonia, and also normal. Here in the left side of the figure, we have some examples of the attention about the model. And we can see that the model, in some cases, pay attention in the background. For example, here, here, and here. And it's important to point the model the correct part that they should pay attention. And with the preprocessing technique that we applied in the images, the model can pay attention in the chest and lung parts of the image. For example, here we have some examples in the part that the model pay attention during the classification. And as last point here about the project, I also worked on classification of retinal OCT images, and also we evaluated bias in the scenario, considering this kind of images. And also I work more recently about fairness. So the main idea of fairness in machine learning is to train the model to deal with different number of images from different classes. For example, we have a specific class that has a lot of images in that case, and another one that has less features, less images in that case. So it's important to model to achieve like some fairness in this problem. And considering the data set that we evaluated fairness, it's about protein location on cells. So it is also a multi-label classification that will apply fairness using these kind of images using convolutional neural networks. So as feature work, My research interests is about machine learning and bioinformatics. And here the two main points is about application of machine learning and bioinformatics to biological data and also to medical data. I think it's important to use machine learning to help people in, with that. I think it's important to understand better biological data and also medical data. So this is the references used in the presentation. And that's it. Thank you for the attention and the opportunity to share my research.
Thank you very much. I'm going to stop the recording now. Okay.